So in this part of the course, we focus on mapping DNA seq-reads. And one of the main applications of DNA sequencing is variant analysis. So you're going to search which variants exists in the population or in a certain individual. So for this application, the mapping is very crucial, we have seen that. So you have to make sure that you're using the right reference for doing the mapping. So variant analysis is mostly done in human. So you know that there are many builds, many versions of the human genome. And for human, using the most recent one, so number 38, is the best choice here. The reason is that it contains the highest number of known annotated variants. And the sequence is based on a mix of many different people with different backgrounds, with different ethnicity, and so on. While the older version, the previous version of the human genome, so number 37, is mostly based on sequences from Caucasian people. For other organisms, it's not always a good idea to go for the most recent version of the genome. When it's very recent, and I think that's the case for mouse, it might be better to go for the previous version. The reason for that is that the annotation is always lagging behind. So when a new genome is released, it has to be annotated. And that annotation process is quite slow, right? It takes a lot of time and effort. So it always lags behind. So very recent versions of genomes very often have less annotation than the previous version. So for these organisms, it might be a better idea to go for the previous version of the genome.